Hi semua. Now we will learn about the deviations from Mendelian inheritance, which refers to some types of inheritance which do not follow the concepts of Mendel. The first type of deviations from the Mendelian inheritance will be about the codominant alleles. This will be referring to two alleles that are equally dominant for a certain type of gene or characteristic. There will be no recessive allele in this particular case. Both codominant alleles are fully expressed even in a heterozygous condition. For example, we will focus on the human MN blood group, in which the homozygous condition for allele capital M or capital M will produce the M blood type for an individual. Meanwhile, the homozygous condition for the allele capital N or capital N genotype will produce the N blood type for a particular individual. Meanwhile, in a heterozygous condition, or the genotype will be capital M and capital N, the individual will be having the MN blood type. This shows that both the capital M allele and also the capital N allele will be able to show their own characteristics in a heterozygous condition. This case involving codominant alleles deviates from the Mendelian inheritance. Why? Because the phenotypic ratio for the F2 generation will not be 3:1. Instead, the ratio will be 1:1. As you can see in this diagram, if we cross an individual with the M blood group with another individual with the N blood group, we will be getting an F1 generation individual which is a heterozygous capital M and capital N genotype. And if we do a self-cross between these F1 individuals, finally for the F2 generation, this type of phenotypic ratio will be obtained, which is 1:1. This is totally different from what Mendel suggested. The second type of deviation from Mendelian inheritance will be about the incomplete dominant alleles. This will be referring to two different alleles which are not completely dominant over the other. In this case, a heterozygote will show another different phenotype which is different from the parental phenotypes. For example, if we look at the color of the flower in the antirhino species plant or the snapdragons, this will be the alleles that will be involved. One of the allele will be this C capital R allele, which is actually the allele for the red flower. Another one will be the C capital W allele, which is the allele for the white flower. If the red snapdragons with the genotype of C capital R are crossed with the white snapdragons with the genotype of C capital W, then all of the F1 generation will not be having any red flower. They will not be having any white flower as well. Instead, all the F1 generation will be having pink flowers, which are different from the phenotypes of the parents. And the genotype will be C capital R and C capital W. This shows that in a heterozygous condition, another phenotype will be produced, and the phenotype will be different from any of the two parental phenotypes. This case involving the incomplete dominant alleles also deviate from the Mendelian inheritance because the phenotypic ratio for the F2 generation will also not following the 3:1 Mendelian ratio. Instead, the phenotypic ratio for the F2 generation will be 1:1. As you can see in this diagram, if we start with a cross involving an individual with red flower with the genotype of C capital R, C capital R, with another individual having white flowers with the genotype of C capital W, C capital W, then what will be obtained for the F1 generation will be snapdragons having pink flowers with the genotype of C capital R, C capital W, a heterozygous type of genotype. And if we do a self-cross between these two F1 individuals, between these two pink flowers, then finally we will be getting the phenotypic ratio for the F2 generation, which is 1 having red flower, 2 having pink flowers, and 1 having white flowers. So the phenotypic ratio is not 3:1. Instead, what we can see here, the phenotypic ratio for the F2 generation is 1:1. The third type of inheritance, which does not follow the Mendelian inheritance, will be about the multiple alleles. This is the case when 3 or more alleles determine the phenotype for a character of a single gene. For example, we will focus on the inheritance of the human ABO blood group, in which for this particular characteristic, there will be only one gene, the gene for the human ABO blood group. However, there will be 3 alleles for this gene. The alleles will be the alleles of IA, the alleles of IB, and the allele of IO. And so the individuals having the A blood type will be having the genotype of IAIA or IAIO. Meanwhile, for individuals having the B blood type will be having the genotype of IBIB or IBIO. Meanwhile, individuals having the AB blood type will be having the genotype of IAIB, and finally, individuals with the O blood type will be having the genotype of IOIO. And if we try to cross an individual with the A blood type with the genotype of IAIA with another individual having the B blood type with the genotype of IBIB, then we will be getting an F1 individual with the genotype of IAIB, and the blood group will be AB. And if we continue to do a self-cross between individuals having the same type of genotype, which is IAIB, crossed with another genotype of IAIB, then finally the phenotypic ratio will be different from what Mendelian has suggested, which is not 3:1. Instead, we can also see the phenotypic ratio of 1:1. And actually, this phenotypic ratio may be different depending on the type of cross that we will be doing. The fourth type of deviations from Mendelian inheritance will be about the polygenes. This occurs when one character is controlled by not just one, but by 2 or more genes. And this shows the cumulative effect of more than one gene. For example, height in humans is one type of polygenic inheritance which may be controlled by more than one gene. For example, we can say that this particular characteristic is controlled by genes A and B. Gene A will be having a capital A allele which carry 1 unit of tall, while the allele of small letter A will be having 0 unit of tall. At the same time, the B gene will be having the allele capital B, which carries 1 unit of tall. Meanwhile, the small letter B allele will be having the 0 unit of tall as well. And so, individuals with the genotype of capital A, capital A, capital B will be having 4 units of tall, and this individual will be very tall. Meanwhile, an individual with the genotype of capital A, capital B, small b will be having 2 units of tall, and this particular individual will be having a medium height, and an individual with the genotype of small a, small b will be having 0 units of tall, and this person will be very short. So this shows the cumulative effect of the genes A and B. Another example for this polygenic inheritance will be about skin pigmentation in humans and also the weight in humans. In polygenic inheritance, environmental factors could affect the phenotypes. For example, an individual who is initially genetically short might become a bit taller due to a healthy diet with lots of vitamins and minerals. And the phenotypes can be measured, meaning that it is showing a quantitative characteristic. And for a population, if we try to do a graph, we will be getting a bell-shaped curve or a normal distribution among the members of a population. For example, if we refer to this picture, we will be able to see that there will be some tall individuals over here, and there will be also some short individuals over here, and in between there will be many individuals having intermediate heights. The fifth type of deviations from Mendelian inheritance will be about the linked genes. This will be referring to genes that are located on one same chromosome. The linked genes do not separate during meiosis, and they tend to be inherited together. For example, in this diagram, we can see that there will be two genes, gene R and gene S, located on one same chromosome. And during the formation of gametes, both genes R and gene S will be carried together into one same type of gametes. Dihybrid cross involving linked genes does not follow Mendel's second law, and as a result, the phenotypic ratio from a test cross on a heterozygote will not be 1:1:1:1. For example, if we look at the inheritance of the body color and also the wing size in fruit flies or the Drosophila species, we should know that the genes for both characters are linked on one same chromosome. For the body color, there will be this dominant capital B allele for grey and this recessive small b allele for the black body color. While for the size of the wings, the capital V allele, which is the dominant allele, will be giving a normal type of wings, while the small letter V allele, which is the recessive allele, will be involved in producing the vestigial type of wings. The vestigial type of wings will be referring to wings that are small and crumpled. And the genotype for the homozygous Drosophila, having grey body and normal wings, will be written this way. If you notice, we write the genotype differently from a normal Mendelian dihybrid. In this case, involving linked genes, we write the capital B allele together with the capital V allele. And so, the genotype will be capital B, capital V / capital B, capital V. Meanwhile, the genotype for the homozygous Drosophila having black body and vestigial wings will be written this way: small b, small v / small b, small v. There are two types of linked genes that we have to know. The first type will be linked genes without crossover. In this particular case, if we do a test cross on a heterozygous individual, this will produce offspring with the phenotypic ratio of 1:1. And this is different from the Mendelian dihybrid test cross, in which the Mendelian dihybrid test cross should be getting the phenotypic ratio of 1, 1, 1, 1. So, if we look at this genetic diagram, we start with the parental generation of Drosophila having grey body and normal wings, and we cross with another Drosophila having black body and vestigial wings. The genotype will be capital B, capital V slash capital B, capital V for this particular parent. Meanwhile, another parent will be having the genotype of small b slash small b, small v. The gamete from this grey and normal wings individual will be capital B, capital V in one gamete. Meanwhile, the gamete from the individual having black body and vestigial wings will be having the alleles of small b and small v. If we combine these two gametes together, then the F1 generation will be having the genotype of capital B, capital V slash small b, small v. Now, if we continue with a test cross on the F1 individual, this will be the genetic diagram that can be shown. And finally, we'll be able to see that the F2 generation will be producing two types of offspring, and the ratio will be 1, 1. One type of offspring will be having grey body and normal wings, meanwhile the other half will be having black body and vestigial wings. The second type of lineages will be lineages with crossover. If we do a test cross on a heterozygous individual, this will produce offspring with the phenotypic ratio which is not 1, 1, 1, 1 or even 1, 1. Instead, we will be getting 4 types of offspring, and there will be 2 phenotypes that will be larger in number. This is said to be showing the parental phenotypes, meaning that these 2 large groups will be showing the same type of phenotype like the parents. Meanwhile, another 2 groups will be in a smaller number. This will be showing the recombinant phenotype, which is different from any of the 2 parents. If we refer to this genetic diagram, which shows a test cross done on a heterozygous Drosophila with the genotype of capital B, capital V slash small b, small v, then we will be able to see that the gametes that can be formed from this individual will be these 4 types of gametes, in which this capital B and capital V will be together and goes into one gamete. The same goes for this small b and small v. They will be linked together and go into one same gamete. However, due to crossover, this capital B allele can be joined to this small letter v and go into one same gamete. Meanwhile, this small letter b can also be linked together with this capital V due to crossover and goes into another gamete. So there will be 4 gametes that can be produced, 4 types of gametes that can be produced due to this crossing over. Meanwhile, for this homozygous recessive individual, which will be having black body and vestigial wings, the genotype will be small b, small v slash small b, small v, and there will be only one type of gamete that can be produced, and the gamete will be having the alleles of small b and small v. If we combine all these gametes from this one heterozygous individual with the gamete from this homozygous recessive individual, then we will be getting the F1 generations, and there will be 4 types of offspring from this test cross. One type will be having grey body and normal wings. Another type will be having black body with vestigial wings. Another type will be having gray body with vestigial wings, and another one will be having black body with normal wings. Even though there are 4 types of offspring from this test cross, the phenotypic ratio will not be 1, 1, 1, 1. Why? Because we can see that there will be 2 groups that will be in high numbers. Meanwhile, there will be another 2 groups that will be low in number. So these two, the Drosophila having gray body and normal wings, and also the Drosophila having black body and vestigial wings, will be showing the parental phenotypes, meaning that this will be the same. This particular fly will be the same like the parent, this parent over here. Meanwhile, this Drosophila having black body and vestigial wings will be the same like this parent over here. So they will be showing the parental phenotypes. Meanwhile, the other 2 groups, which will be low in number, will be showing the recombinant phenotypes. Why? Why recombinant? Because the phenotype that they are showing will not be the same like any of the 2 parents over here. The last type of deviation from the Mendelian inheritance will be about the sex-linked gene. This will be referring to the case where the gene is located on the sex chromosome. And usually, the sex-linked gene will be located on the chromosome X in humans. For example, the inheritance of hemophilia in humans involves the sex-linked gene. Hemophilia is a disease in which the individual having this disorder will not be able to stop bleeding. This is due to the recessive allele small h, which is located on the X chromosome. If a person will be having this type of genotype, which is X capital H, X capital H, this person will be a female, and this female will be a normal person, meaning that the female will not be having any hemophilia. Meanwhile, if the genotype will be X capital H, X small h, then this female will be a carrier, in which this female will not be having any hemophilia, but this female will be able to pass the recessive allele of hemophilia to her children. And if the genotype for this person will be X capital H and Y, then this will be a normal male. However, if the genotype will be X small h and Y, then this will be a hemophiliac male. Usually, hemophilia occurs more on the males compared to the females. Another example of a sex-linked gene inheritance will be the color blindness, which also occurs more on the males compared to the females. This diagram shows the inheritance of hemophilia in humans. The father is a normal male with the genotype of X capital H and Y. Meanwhile, the mother is having the genotype of X capital H, X small h. This means that the mother is a carrier. From these 2 parents, this will be the possibilities of the children, in which they might get a son, a normal male, without any hemophilia, if the genotype is X capital HY, and they might also get a normal daughter with the genotype of X capital H, X capital H. However, they might also get a daughter which is a carrier if the genotype is X capital H, X small h, and they might also get a son which be having hemophilia if the genotype is X small h and Y.